Hello class, I hope you're doing well today. We're going to continue on with our evidences for evolution. So let's get started. So last class we talked about, in terms of evidences for evolution, artificial selection, how humans can model this process of selection and how organisms can change in response to selection pressure. And also the fossil record, these remains of ancient organisms that show us how life has changed over the history of life on Earth. Today we're going to continue on with those evidences for evolution. So one line of those evidences is going to be comparative anatomy. So when we look at comparative anatomy, one of the types of traits that we look at are what we call homologous traits. Now, homologous traits or homologous structures, these are structures that have the same evolutionary and embryonic ancestry, but have different functions. So they came from the same ancestors, and when we look at them in embryos, they develop from the same structures, but realize they may indeed have different functions as they've been adapted to different purposes. And these homologous structures are evidence for common ancestry, or a shared ancestry amongst organisms. So let's take a look at some examples here. Here we're looking at the skeletal structure of a human arm, and last class session we talked about the humerus, the radius, the ulna, the wrist bones, hand bones, and finger bones. So one bone, two bones, and then bunches of little bones. If we look at, say, a shrew, which also uses its front arms for grasping, we see those same bones present. One bone, two bones, bunches of little bones. Well, let's consider an animal that uses its front limb very differently, say primarily for walking, like, say, a dog. When we look at the front limb of a dog, we see those same bones present. The one bone, the humerus, radius, ulna, wrist bone, hand bones, and finger bones. But they have just been selected for a different purpose, primarily for locomotion. Or let's take a look at, say, the leg of a horse or a sheep. Here again, we see those same bones present. One bone, two bones, bunches of little bones. After that, wrist bones, hand bones, finger bones. But what's really interesting here is that when we look at a front leg of a sheep, what we might actually think of as the knee is actually made up of the same bones that would make up our wrist. Now, if we think about that, having a bunch of bones forming in a joint, is that likely to be the best arrangement for bones in a joint? Probably not the best arrangement, especially for a weight-bearing joint at this point. However, keep in mind, evolution does not select for what is the best. Evolution selects for what works. And so certainly the horses and sheep are very successful, and this has worked out well for them. Now let's consider another application. So animals that swim. So if we look at the front fin of a dolphin, when you look at the front fin of a dolphin, it actually is very similar to the front fin of, say, a shark. It is a rigid structure. It does not bend very much, and it is designed to help maintain the proper orientation as a high-speed aquatic predator, just like a shark's fin is a rigid structure. Now, unlike a shark's fin, a shark which is supported by rays of cartilage, or a fish's fin which is supported by rays of bones, when we look inside that dolphin fin, what do we find? We find those same sets of bones that are in your arm or the leg of a dog. So you have one bone, two bones, bunches of little bones, humerus, radius, ulna, wrist bones, hand bones, and finger bones. Why do they have these bones? Well, this is probably not the best arrangement of bones, plus having the 5 finger bones present there. The reason they have these is best explained because that their ancestors had those sets of bones, and they were selected for that shorter limb that was very rigid and not really bending. If we look in the limb of a seal, again, we see similar characteristics, which really make those limbs best suited for swimming. So the limb of a seal would be very similar in function to, say, the wing of a penguin, for example. We can even take a look at adaptations for flight. So again, whether we're looking at a pterodactyl, which is a reptile that you would fly, a bird, which we know flies, and bats, we see those same things. So in the wing of a pterodactyl, the flight surface was actually supported by a layer of skin between the pinky finger and the body. But again, we see one bone, two bones, bunches of little bones. In the birds, those bones have been greatly reduced to save weight. So you have one bone, two bones, and significantly fewer bones here. But the flight surface is supported by the feathers. In a bat, it's actually webbing between all of the fingers that allows those animals to maintain flight. So all of these skeletal structures have the same evolutionary history, and that they come from that same amphibian type ancestor that came out of the ocean with one bone, two bones, bunches of little bones, but it's been adapted to different functions. And these are great examples of homologous structures. Now, another type of trait that we might talk about are analogous traits or analogous structures. Analogous structures have the same function, but have different evolutionary or developmental origins. And a great example we might look at here is the wing of a butterfly compared to the wing of a bird. Now, a butterfly wing, you have this exoskeleton, which provides the support, and the flight surface is actually covered with tiny scales. There is not an internal skeleton like we find on the bird, and these structures develop from very different origins as they develop embryonically. So, however, they do serve the same function. They have the same function, and that function is flight. So what analogous traits point to are what we call convergent evolution. These are organisms that have evolved similar traits because they are dealing with similar selection pressures. Another example we might see with this is if we compare the body design of a shark to that of a dolphin. So both of these have rigid pectoral fins. They have this dorsal fin. In fact, you might be out at the ocean and see a dorsal fin coming up, not knowing whether that's a dolphin or a shark. And those are structures that help those organisms be very good, high-speed aquatic predators. And so they have similar structures because they have similar selection pressures. They're adapting to similar environments. And that is convergent evolution. Another line of evidence we can take a look at is comparative embryology. In comparative embryology, we see that we have similar patterns of embryonic development that diverge as development proceeds. So if we look at all vertebrates, animals with backbones, they all start out with a single egg, fertilized egg that develops eventually into a solid ball of cells and then a hollow ball of cells, eventually later on developing into this folded structure called the gastrula. We'll learn about those a little bit later on. But if we look early on in the embryo, what do we see? Here we've got eyes that are present. Kind of the brain is developing here. But all of these vertebrates are going to share these structures near the neck called pharyngeal pouches. So it doesn't matter if it's you, a chicken, a snake, a salamander, a fish, your dog, what have you. we all had these pharyngeal pouches early on in development. Now in the fishes, those develop into the gills and also into the jaw structure. In the salamander, as animals came onto land, those structures started to be repurposed. As gills were not as required on land, those structures were selected to be able to detect sound waves. And so nowadays, those structures develop into things such as the bones of our inner ear and the likes. We also all have a tail. So at 5 to 6 weeks of age is when your tail was the longest. You indeed had a tail early on in your embryonic development, and that links us back to our ancestors, which did indeed have tails. Now, let me ask you something. Let's go back here, take a look. When we're looking at these embryos, once we get up to the land vertebrates, we see that they have front limb buds and hind limb buds. If we were to look at, say, a dog embryo, front limb buds, hind limb buds. A turtle would have front limb buds and hind limb buds. Even a chicken, we don't really see that right here in this embryo, but they would also have front limb buds and hind limb buds. What about a dolphin embryo? If we look at a dolphin, they do not have hind limbs. But the question is, will they have those hind limb buds that were present in the human? And as we look at this embryo, what do we see early on in development? Right there is here are the front limb buds, there are the hind limb buds. So this is evidence that their ancestors had hind limbs. So this embryology really can help us to trace the relationships and ancestry of organisms. Now, as these embryos develop, eventually those hind limb buds are reabsorbed so that they do not have hind limbs, which makes them much more agile and swift aquatic predators.